Welcome back. It's Friday morning on London Calling 2023 and people are starting to file back into the building. We had Clive's tech update last night. People went out for dinner, went out for drinks. There's a few husky voices in the building, but we're really excited for another day of wonderful science lined up. And I'm joined in the studio here today by 3 attendees of London Calling to come and share their views on the science from yesterday and what we've got on the day ahead. So thanks very much for joining us after a late night and a busy day. First of all, I'm joined by Tip, who's here from Bangkok, all the way from Bangkok, and a wonderful pioneer of genomics in cancer. So I want to ask you, Tip, in your environment and the work you're doing in Bangkok, where are the bottlenecks for genomics making an impact in cancer? And is there anything that you heard yesterday that makes you optimistic about bursting through those bottlenecks?
Okay, thank you for the question. For in Bangkok, people now can use nanopore, can run nanopore sequencing. But the problem is about the bioinformatics, the data analysis. And the talk on yesterday, Clive talk, he mentioned about the automatic analysis. This is so cool. Yeah, I think if we can use that software, EP2Me, and pipeline, then we can solve that problem. Yeah.
And what does that mean? Who will have access to sequencing in genomics who previously may not have been able to access it as a result? What does that look like in the context of your ecosystem?
So actually, it's not only in Bangkok.
No.
Everywhere in Southeast Asia, in Malaysia, Indonesia, they always keep asking about how to analyze the data. Yeah, that's why I think bioinformatics part is very important. And now I think we can use EP2Me to solve that problem. Yeah.
It's so fantastic to hear because the teams are really focused on supporting that translational research community, the people who are working so hard to translate the discoveries that are being made with Nanopore into formats where we can bring those insights into everyday use. So I think the Epitome team at home will be really pleased to hear that. And actually Abdul, who is sitting right where you are yesterday, was saying that the Epitome improvements have allowed him in the Gambia to move from bringing people into his building to train them to being able to train them remotely. So I think the Epitome team will be really pleased to hear that. So thank you so much for sharing that. Okay. So John has joined us, I think the first teacher that we've had here at London Calling as a delegate and indeed a presenter. So upstairs we have a STEM day happening. We have 60 students from non-fee paying schools all around the UK, and they're here being exposed to genomics, to careers talks, and in fact some very, very special sequencing and some specific objectives, some experimental goals. So John, would you like to tell us how you're what you're asking the students to do with you today?
So we're doing the whole pipeline today. They're going to be getting the DNA out of some leaves from daffodils that have been kind of sent from a national collection up in Scotland, as well as a little bit of a curveball. There's one from Cornwall as well in there. And they're going to get the DNA, prepare the library, and hopefully get the sequencing started. So over the weekend, maybe, we'll have 10 new chloroplast genomes ready for assembly, as it were, hopefully. And then we can maybe get these young people as co-authors on a scientific paper from today.
I've always thought that I would like a front cover Nature paper by a 14-year-old. Do you think that's achievable?
There's some amazing science out there. There was a school child is like this, who's I think 18 years old, that sequenced his tropical fish.
His angelfish.
Yes. That had died in his name. There's a couple of schools in Chile that have sequenced little hexapods that they find in the soil in their playgrounds. There's amazing scope for making completely new to science knowledge using Nanopore. It is just phenomenal, and it's inspiring every time I look at it and I find something, I stumble across something, and I just like, wow, I want that.
You are touching on something that I think is incredibly close to all of our hearts. So I think we all see the world has many challenges today, whether that's health, environmental, food security, and the communities that are affected by those challenges are not necessarily the ones that currently have the genomics infrastructure. So I think at Oxford Nanopore, we are keen on developing platforms that are more accessible so that the communities who have the challenges can also be the communities who solve those challenges using genomics. And we have to do that by starting with genomics understanding and life sciences understanding from that high school level up. So there is much more to come on education, and we'll touch on Clive's talk in a minute. But I'm going to move to Matt Luce. Hi, Matt, who's in from Nottingham today. Hi, Matt.
Hi there.
So Matt, as you may know, anyone who's in the Matt Luce following zone, the fanzine, as we were discussing earlier, a real pioneer of promethine in particular, and work on promethine and also with adaptive sampling. So in your work at Nottingham, what's exciting you right now, and what's excited you yesterday from the science that you saw in the room?
I mean, it was fabulous to see so many different things happening, from the small scale we've just been hearing about, small scale in terms of being able to do things easily, to the massive high throughput projects that are starting. Tens of thousands of genomes being sequenced a year in multiple different locations. It's just really, really exciting to see that, I think. With respect to the Promethean, the thing I'm most excited about now is the new smaller devices. So we've just received our P2 Solo. I actually thought about skipping the conference entirely and just staying at home and using that, because it's fabulous. You can generate large amounts of data, hundreds of gigs, locally where you are, without the need for all the huge infrastructure, and that's going to be transformative again, I think.
Why did you not bring your... There's the voice of God calling people in. Excuse us for a second. Why did you not bring your P2 with you?
I seriously thought about bringing it with me because it would have been fabulous. I was a little bit worried that I might leave it somewhere, and it might look a little bit...
You might have lost it in your bag like Clive did yesterday.
Exactly, that's the problem. So, I'm looking forward to getting back to the lab. I genuinely am. I want to plug it in, switch it on, and run some libraries. And actually, the important thing about that, we want to get that into the clinicians' hands where we are as well. So we're really looking forward to seeing how that works out.
Well, this is a really serious point, because we talk about promethine, and traditionally high output sequencing has always been associated with centralized sequencing centers where the samples have to travel to the lab. And that sort of reduces the number of institutions and people who can have access to high throughput, high output sequencing. So one of the things we've been trying to do with P2 is break the paradigm of centralization by enabling high output, large number of plant genomes, human genomes, and bringing that to anyone who needs it. So it sounds like Matt's excited about that. Tip, is that something that excites you as well?
Yeah, so actually I just got P2 Solo and I ran 3 samples. The first one is metagenomics, and the second one is 88 bacterial genomes, and the third one is human cancer. So I confirmed that within 5 days we can get the data with P2 Solo, and I hope P2 will come soon.
Very soon, as you heard. P2I is coming right now. And so, what does that mean if we've got technology that allows high output, say cancer sequencing, in a decentralized device? What does that mean in this sort of translational future healthcare environment? What do you think that means across the Southeast Asian network, for example?
So I think combined with the bioinformatics, then we can know the location of CNV, copy number variation, SNP, and pathogenic SNP, for example. And then can be translated into clinician, and then they can use that data to choose the treatment option.
So talking of copy number variation, we were talking about the brain samples that you've been doing in Nottingham. So how does that relate to the work that we heard, say, for example, Clive talking about last night, the work that you're doing in those difficult to sequence brain samples?
Well, I think it's going to be fantastic to see how we can improve that work, and time to answer in some of these experiments is so important. The point you made about the large centres, it always depends on your goal, doesn't it? What you're trying to achieve, and for a long time the only way to do these things have been in large centres. But imagine you had something, brain tumours are an example, where you might need to know something like methylation status. And that will have a key impact on treatment for those patients in the future. Depending on where you are right now, the turnaround time for that result can be anything up to 6 to 8 weeks. Sometimes it's a lot closer, and that's just for us here in the UK. So now we can take that, maybe get it down to a few days. Ideally, I'd like to get that down to hours and give those results. And I think that's going to be super exciting.
I think we can agree that the patients and the families would also like to see that change, so that we could have better care and better emotional journeys through these healthcare journeys in the future as well. So finally, one last call for John before we go. So Clive talked about a low power, much lower cost ASIC for the future of our devices. It's not happening tomorrow, but our vision is anyone, anything, anywhere. What does that mean for you as a teacher, do you think?
I want to be able to do this, and I want to continue doing my little sequencing, but much more important for me is thinking about those future scientists, thinking about the people that follow Mac, it's about inspiring the next generation, and we have to bring as much as we can into the classroom to give students that really fully informed decision whether science is for them or not. And sequencing has to be a part of our future science education going forward.
Well, I think we're about to start. What I want to see, John, next year is one of your Daffodil students from today right up there opening on Friday morning. Perhaps we can set that as a goal, shall we?
Shall we?
Let's do it. Okay, have a great day today. Thank you so much for joining us.
Thank you so much.
And we're over to Rosemary to open the talk on Friday morning. Have a great day, everyone. Bye.